Alright, so we're going to do round 2 of this. Sorry I'm not there, and I apologize in advance if I cough into the microphone. I am sick. Hopefully we'll be in on Tuesday. If not, I'll be in on Wednesday. So for today at least, you are going to do the video set of notes again. We're going to go over transcription. You guys are going to do an online tutorial. I'll show you how to get into there and how to go through that. You're going to work on your homework then, and then your exit ticket. I have it up in the Google Classroom that if you finished all of this already, you can work ahead on your science fair project. So meanwhile, you can pause this. You can take as much time as you want. You can copy down these questions, your ICANs, and your homework for your catalyst sheet. If you do not have that paper, sorry, make sure you get that from the sub. But other than that, we are moving on. So you guys are experts on DNA already. You know that DNA lives in the nucleus, but the information that's in that DNA and all those C's and G's and T's and A's has all of the instructions for making the proteins in our body. And the central dogma of biology says how DNA is converted into RNA, which is converted into proteins. So DNA into RNA is what we're going to be talking about today. How we go from DNA to RNA is a process called transcription. So RNA itself, it is another type of nucleic acid. Sorry. It is RNA, not DNA. There are three main differences that we're going to talk about. The nucleotides have a ribose sugar, so that's why it's RNA instead of deoxyribose, which is DNA. In RNA, we do not have thymine anymore. We have A, U, C, and G. It's the normal base pairing rules, except A goes with U and C goes with G. And now it is single-stranded. So DNA, think D for double. RNA is a single-stranded molecule. And we have three different types of RNA. And we'd say which ones they are based on the little tiny letter in front of it. So if you look over here, mRNA is short for messenger RNA. So it's just like a messenger. It carries the information from the DNA into the ribosome. Ribosomal RNA down here, that's little r RNA that makes up part of the ribosome. And then down here, the tRNA, that stands for transfer. It is going to be transferring an amino acid to the ribosome, and it's going to bind to the mRNA. So you can see these right here are going to match up perfectly with the mRNA here, whichever one has the correct base pairing rules. Now DNA is always, always, always in our nucleus. That's because it has a ton of information. It's way too big. It cannot get out. But mRNA is small enough to leave the nucleus. So that's why it's called the messenger. It's going to take the information from the DNA. It's stored in the nucleus, and it can leave through those nuclear pores, those itty bitty holes right there. So this is actually the process of transcription. So when we copy DNA to RNA, specifically mRNA. Now transcription, you can see we are going to have more enzymes like we talked about before. So in this case, it's RNA polymerase instead of DNA polymerase that we talked about before. RNA polymerase is going to open up those hydrogen bonds, and it's going to use it as a template. So just like we did before, it's going to use those complementary base pairs to make a copy into mRNA. The mRNA can then leave the nucleus. It has all the information that it needs, and then it goes to the ribosome. And if you forgot, the ribosome is where we're going to make proteins because that's its job. But as of right now, we're just talking about how we make mRNA. And like I mentioned before, RNA has U instead of T, so we still have more or less the same base pair rules except for one difference. So if you have A in DNA, it's going to be U in RNA. Everything else is going to be exactly the same. C still goes with G, T in DNA still goes with A in RNA, and C goes with G. This is the only one that's different. A in DNA goes with U in RNA. So if we had this DNA right here, if you had to say, we are doing transcription, we are turning this into RNA message, what would it be? I'll give you a couple seconds to think about that. Write it down on your notes. Sorry. So if this were your RNA message, T goes with A, and so on and so forth. Notice that A is being paired with U, but everything else is the same. Try it one more time. The sequence of mRNA made from this DNA, AACTG, doing transcription, turning it into RNA, you would get this messenger RNA. So again, A goes with U, everything else is still the same. And that's all that transcription is. So once you have all these notes copied down, feel free to rewatch this if you need to, but I'm going to show you how to open up the transcription tutorial properly. So if you come into the Google Classroom, I'm using period 1 as an example. You come down here, this is the first thing on your stream. So Monday's class, it says, okay, you watch the notes. I'm going to add the link right here next. The transcription tutorial, you have to follow these specifically. So you're going to click on this PowerPoint right here. You can't do it just like this. You have to come up here to the 3 little dots, open in a new window. And then what you're going to do from here, you're going to click this button right here, which is download. It will download. You will open it up. Some computers are faster than others. And then what you can do is come up here to slideshow and play from start, the very first one. And all you have to do is use your mouse. You're going to start from the beginning. You're going to follow all the questions, answer the questions that go along with it. You can always go back to home right here. But other than that, that is all that you are doing. Follow the questions, answer what they say. After you finish that, you're going to look back here. It's going to tell you you are going to do your transcription practice. It's the next 2 pages of your homework that are due tomorrow. And then you have your exit ticket. All of these papers that you have, you can give straight to the sub. We're not going to be in the classroom for the end. I will get them all when I come back. Like I said, ideally on Tuesday, if not on Wednesday. Be good, and if you're good, if I get good notes back, I will definitely bring in brownies like I did last time. Other than that, if you have any questions, send me an email, send me a remind message, and I can get that back to you as soon as I can. All right, be good.